So it is 12 o'clock. Welcome to the second virtual BioM Lunchtalk on cell, gene, and Immuntherapy. And my name is Ralf Huss. I'm the Managing Director of BioM, and I'm very pleased that we have two distinguished speakers on this topic today who present their technologies and their companies. The first presentation will be given by Dr. Karin Wiskirchen from SCG Cell Therapy, and the second one from Dr. Takis Atanassopoulos. Excuse the wrong spelling maybe of your name. It doesn't come easy off my tongue, so sorry for that. Vice President of Gene Therapy and Research and Vector Technology of Vigeneron GmbH. So we are very pleased to have you here. And for those of you who have already participated in such lunch talks, you know that you can ask your questions in the chat, which I will read out loud and ask the presenters to answer the questions. The presentations will be about 20 minutes each. I will go on mute and will turn off my camera, and I will reappear if it comes to an end of the allotted 20 minutes of the presentations. So once again, thank you very much, everybody, for joining and participating on this great topic. And let me introduce Dr. Karin Wiskirchen. She studied biology in Marburg and Gießen with a focus on virology. During her PhD thesis at the Institute of Virology at the TU München, she focused on development of CAR and TCR-based T-cell therapy against chronic hepatitis B and HPV-associated HCC. Just as a remark in between, having been a clinician for many years and until last year, I know this is still a very burning issue. We are really looking forward to hopefully see a solution or clinical solution and therapy on this topic. So the last sentence on this is together with the company SCG Cell Therapy, where she's now employed as managing director of the German research arm. The project has made the translational leap into the clinic, which makes it even more important to everybody. So welcome, Dr. Wiskirchen, and I'm sure you will tell us a little bit about the company and about your technology. The stage is yours.
Yeah, thank you very much. First of all, to BioM for organizing the event and also for inviting SCG to present. Thank you, Dr. Huss, for the introduction. And actually, you might not remember this. We have met some time ago. I think it was 2012 in Erlangen at the Forum Cell-Based Therapies, where I presented my PhD topic on a poster. And you came by the poster and we discussed it, and you wished me good luck with the project. And never would I have imagined at that point that 11 years later you would introduce myself and the company to present data. So it feels really good. This was actually also the project on which SCG Cell Therapy was based on when it was founded. So what is SCG Cell Therapy? We are a fast-growing biotech company which covers the entire value chain of cell therapy development, meaning target discovery, product development, GMP manufacturing, vector manufacturing, and the clinical rollout. We are founded and headquartered in Singapore, and SCG actually stands for Singapore, China, Germany, focusing on the treatment of infectious diseases and its associated cancers. And our lead product is the before mentioned TCR T-cell modality for treating HPV-induced HCC. And for this, we already have first clinical data. And for each of those points here, I would like to go a little bit more into detail. So in Singapore, we have have one production site, but it's more important as a financial and management site. It is also kind of a bridge between the Asian and the Western countries. Then we have China, where we have large-scale manufacturing. and also the clinical trials happening now, or starting there at least. The reason is that the product that we have as a lead product, the HPV-induced HCC as an indication, is very prevalent in China and really a huge burden that needs to be treated. So it's very easy to find patients to treat there. And also China has a special kind of regulation that in cell therapy, you would always start doing an IIT before you are even allowed to start a phase 1. So this helps you to get started in translating your projects. And then we have Germany as an R&D site, and we chose Germany for this because historically the know-how comes from Germany, and also because of the access to well-trained staff in Germany and better access to academic collaborations and materials. And to be more precise, so the R&D site is in Planegg at the IZB, where we established our TCR isolation platform and use it to feed our global pipeline. And then in China, we have 3 different sites, or even actually it's 4. So in Dalian, we are currently building a production site for cell therapy products. It's pretty huge and will be able to produce around 350 products a year. And it's supposed to start by end of the year. Then in Shanghai, we have 2 different sites. One is for the currently ongoing GMP production and the QC, and the other one is for translational and IND enabling studies. And then in Guangzhou, we have the vector production. And in the last week's seminar, it was mentioned that vectors are very expensive and are really a nuisance in cell therapy. So we have, as a solution to this problem, acquired a company that was back then in Guangzhou, so that we have now an in-house production of the vector. And this makes it fairly easy because we have very short ways of communication, and also the costs are really low now for us. And then in Singapore, we are also building a GMP lab at the moment that will be opened in July. And from this, we are supplying the clinical study sites in ex-China, meaning US, Singapore itself, and Hong Kong. So who are the people behind SDG? Our scientific founder is Professor Ulla Protzer from the Helmholtz Center and the Technical University of Munich. She is the director of the Institute of Virology. And I think most of you know her probably from her advisory role during the pandemic. But as a virologist, she's even more renowned as a specialist in hepatitis B research. And she has been working on the cell therapy project for like 20 years now, so even before I started my PhD thesis on this topic. And then we have Dr. Wolfgang Uckert, who is now a retired professor from Max Delbrück Center, and he's a specialist in immunology and vector technologies. Then we have Ke Zhang, who is our chief scientific officer. He also did his PhD thesis with Ulla Protzer. So we actually started on the same day in 2007, and he was focusing more on the molecular biology of hepatitis B virus. And he then went to J&J and became a site head for hepatitis B research development. Further staff are our CEO, Frank Wang, who is a businessman that was the former CEO of Biosyngene, also biotech startup. Then Christine Ma, who's the clinical lead and has developed several clinical products already. And last month, Lang Chi joined us as a CFO, and she has ample experience in bringing small biotech companies to IPO. Then we have Liu Ha, who is the CTO, and he's the founder of Transfer Genes, so the company that we acquired for the vector production. And we have Susanne Wilde, who is with me at SEG Germany and is leading the preclinical research team. Now I come to what we are doing and more importantly, why we are doing it. So SEG's mission is to target and target infectious diseases, pathogens, to prevent and cure cancer. And actually from the cancers that are caused by infection, most of them are caused by Helicobacter pylori. Then there are several viruses contributing to cancer, and in the end also some parasites. And in total, it's around 2.2 million cancer cases per year that are caused by an infection, so quite a lot. And if you look on the right-hand graph, you see that in purple, that's purple, that's Asia. So around 2/3 of these cancers are diagnosed in Asia, and the dark purple part is here is China. So over 1/3 of them are detected in China. So it's a real high unmet need there, especially in Asia, and it has not been the priority. It has been underappreciated by Western companies so far. SEG is focusing on 4 of these top 5 infection-related cancers. So for gastric cancer, we are working on something against Helicobacter pylori and EBV. For cervical cancer, we have a target from HPV, human papillomavirus. Then against liver cancer, we have the HBV, hepatitis B virus targeting T-cell receptor. And for head and neck cancer, we have papillomavirus and EBV specific T-cell receptor therapies. And although we call ourselves SEG Cell Therapy, we have broadened our scope a little bit, including also other immunotherapies that modulate the cellular immune response to a pathogen. And this is because it's always a trade-off between safety, efficacy, and cost. And at first, when people are only infected and at a low risk, you might want to use something that is a little bit milder as a treatment and not start with cell therapy. So something like a therapeutic vaccination is warranted, and we are currently developing this against Helicobacter pylori. Then infection might persist over years, causing more and more problems for the patient, and then something like bispecific antibodies become a viable option. And we have some against chronic hepatitis B that we are developing. It's also licensed from the Helmholtz Center Munich. And as a last resort against cancer, the cell therapy comes into play, and this is what I want to focus on today. This is our pipeline. So the lead product is called SEG-101. This is the HBV-specific T-cell receptor carrying T-cells. It is currently in phase 1. And then, as mentioned before, we also have already T-cell receptors against papillomavirus and Epstein-Barr virus for treatment of cervical cancer and nasopharyngeal cancer. And while the HBV-specific TCR is still originating from the Institute of Virology, the HPV and the EBV T-cell receptors were isolated in our SEG site in Germany, so at IZB, where we established our T-cell phishing platform. And this TCR phishing platform is based on a very thorough analysis of the target epitopes beforehand. Then we have high throughput steps to isolate functional T cells. So we are very much interested in knowing the functionality of a given T cell before we isolate the TCR. And this is something where we have a different strategy than other companies who might just use multimer binding and then isolate a T cell receptor and later find out what the functionality of this T cell receptor is. our platform gives us high affinity TCRs and no further maturation steps are necessary. So with the lead product, now I come to the production process in general, which we have established for the lead product, SCG-101. So as mentioned before, we have an in-house lentiviral production, and the lentivirus is pretty good and gives a high purity and high viral titer. Then the steps are quite standard for producing the cells. So we start with the leukapheresis, then we isolate the T cells with the Clinimax, have the lentiviral transduction and expansion, and then harvest the cells and do the QC. The process is almost completely automated and, of course, fully enclosed. So there's not too much hands-on time, and the protocol has been optimized to yield around a 200-fold cell expansion within 10 to 12 days. And the process has been approved by agencies in Singapore, US, and China. So for our lead product, SCG-101, the mode of action is that this HBV virus antigen, it's called HBV surface antigen, is not only present in infected cells, but also in tumor cells in which the virus has integrated. And then it is presented on MHC molecules. And when the T cell that we inject, that is carrying the T cell receptor specific for HBV, can recognize the presented peptide, the T cell will become cytotoxic and kill the target cell and also produce inflammatory cytokines. So of note, it's known or anticipated and wanted that this will happen with both infected cells and tumor cells at the same time. If the patient is willing to do so, a biopsy can be taken before treatment, and that is stained for this tumor virus antigen in the tumor, which you can see here on the left-hand side, which is a tumor that is in this case full of HBs antigen. So in this IIT, as I mentioned before, you start with an IIT in China. We could create some clinical data already. We treated with 3 different doses, and these are 2 examples from the doses of 5 × 10⁷ or 1 × 10⁸ TCR positive cells per kg. And what we see in, I would say, 90% of the patients is really a very rapid increase in ALT. So this is the green lines here, meaning that liver cells are being killed and the ALT is released in the serum and becomes measurable. And at the same time, the viral antigen, HBs antigen, is decreasing. And we see reductions of more than 3 log-fold. And by the time the antigen is gone, also the liver function normalizes again. But this antiviral activity is only a secondary endpoint. Of course, our first goal is to target the tumor. And here I present data from a patient in which we saw a good partial response. So if we look via IHC, we see before treatment, there is lots of HBs antigen positive cells in this biopsy. And 3 months post-treatment, these HBs antigen positive cells are gone. This is actually a liver biopsy. But what we can appreciate here is that the liver is not only carrying infected cells. So actually in these end-stage patients, it's only around 10% of infected cells. But what we see is these areas of HBs antigen positive cells. And these are pre-malignant cells where the virus has already integrated, and these cells are prone to become a tumor themselves. And by CT scan, we also saw that two target lesions that we were focusing on. Here, for example, in this one, present before treatment, and then 4 weeks after treatment, they become darker, indicating that necrosis is happening. And 4 months after treatment, they are hardly visible anymore. And that reconstituted to a partial response with around 70% of tumor response. Our next step is to finish the phase 1, which is at the moment including 3 to 6 patients per dose level. And then by the end of the year, decide on the dose level for phase 2 and start with the phase 2 part of this study. In the end, I also want to talk a little bit about TCR therapy in general. So most progress has been made with CAR constructs, but they have not been as successful in treatment of solid tumors. And the reason is possibly the hostile tumor and microenvironment, which is maybe not as intensive as in liquid tumor, not as intensive in liquid tumor. Yeah. Then that the mere tumor mass is just higher. That has to be attacked, and if you choose a certain antigen with a CAR, you are restricted to surface antigens. And in the space of TCR, T-cell therapy, you can also target tumor-associated antigens and tumor-specific antigens like the viral ones or mutation-associated neoantigens that are present intracellularly. And in our case, we also have a chimeric antigen receptor in hand for the HBV surface protein, and now had to focus on one due to financial constraints, and we saw that in our hands, at least, the T-cell receptor is much more sensitive than the chimeric antigen receptor. So around 1 or 2 years ago, there were not so many companies focusing on T-cell receptor carrying TCR products. And SEG was actually the only one having approval across different continents, so in the Chinese NMPA and the US FDA. And yesterday I looked again at clinicaltrials.gov, and within this 1 year, I think there have been lots of changes. So there are several companies in Europe that are also starting clinical trials now. In the US, they were a lot, but especially in China. Yeah, these are all, I wrote Asia, but it's all China. There's a huge interest in this topic, and many companies emerging and doing clinical trials. Yeah, and with this, I'm at the end, and I would be happy to take questions.
Thank you, Dr. Wiskirchen, for this great presentation. I have noted, or I've written down many questions. And first of all, I'm very glad that I did not discourage you 10 years ago, but rather I wish you all the best and good luck. So this, I'm really pleased to see this. So I don't see any questions in the chat. So let me remind all the members in the audience, you can write your questions in the chat. It's entirely anonymous. I will read them to everybody and to Dr. Wiskirchen. But let me start first, maybe with a couple of naive questions. My immunology time is a little bit back, and what we have seen in the past is that usually MHC molecules are downregulated in solid tumors. And if I understand this correctly, your therapy also requires MHC restriction. So do you also see this in HCC or in these models?
Yeah, so in HCC, the data published has been quite contradictory. So there are publications that say There's no MHC expression at all. There are other publications that say there is enough. So in the end, it left us with taking the biopsy materials and also some purchased biopsies from HCC samples and just stain them ourselves with an HLA-A antibody. And it turned out that I think it was around 90% of the humans had really a good expression. What happens though is that you can distinguish within the tumor. You also see different other cells that are maybe coming from stroma still or from the endothelium, and in those cells the expression is higher than on the tumor itself.
Thank you. There are now questions in the chat or in the F&A part. One question is, and I have written down the identical question. So what is your strategy for the European market? And is there one? And what is your assessment of the European market? I also made the comment, are there enough HCC cases here in Europe? Is that one of the reasons why I'm not going to Europe? But this is a very valid point. So maybe you have an opinion on that.
Yeah, so I mean, at the moment we are in Series B funding, so there's just not enough money to go to all markets at the same time. And if you have to make the choice between US and Europe, unfortunately, US just makes more sense because one, they have more patients than in Europe, and two is that also the process is much easier because the FDA is much more experienced with these kind of cell therapies. And in Germany, especially, we calculated that it would be definitely less than 100 cells per 100 patients per year that could be treated. So it's really a few patients. And even in the US, it's a couple of thousands, but not a lot. So it would go into the area of orphan drug designation.
And so you did not seek advice yet at the Paul-Ehrlich Institute, did you?
Actually, in 2013, when I was still with Professor Protzer, we went for a scientific advice meeting and got some ideas and was quite constructive. But with SCG, we have not gone for one to the Paul-Ehrlich Institute yet. No.
Okay. So there are now more questions. How large is the scale of your clinical phase 1?
So it's 3 dose levels, starts from 5 times 10 to the 7, then 1 times 10 to the 8, and 2 times 10 to the 8. In the IIT, we had also 1 times 10 to the 7 per kg, but for phase 1, we were allowed to skip this dose because we already found it was safe. And so we have for each of these 3 dose levels 3 to 6 patients. So minimum 9 patients in total, and maximum would be 18.
And along those lines, I think this fits nicely, is how do you ensure no off-target toxicity?
So here we have to distinguish a little bit. So off-target in cell therapy means not targeting the HBS antigen. And this we have ensured by testing the T cells against different primary cell lines and tissues that we generated from iPSC. And we did not see any off-target activation. And also we have tested against a library of similar peptides that are similar to our HPV peptide, and also did not see activation. I'm not sure whether the question is referring to off-target or maybe rather on-target of tumor, because a lot of people want to talk about targeting the infected hepatocytes and what this might.
I mean, it says off-target, no off-target activity, as we see sometimes for other antibodies. For example, what we know from trastuzumab, that it gets us, cardiac failure or at least side effects. Yeah.
Yeah, and I mean, we are quite happy because we target a viral antigen, and it's very unlikely that there's something similar in the body because the T cell receptor we are using is a fully human one. and it's taken from a donor that has cured HBV infection and developed a strong T-cell response. And in this donor, of course, there was a natural selection. So you would assume that no similar self-antigens can be targeted.
The next question is, I think, very much along the lines that I was trying to ask initially. It says one common immune escape mechanism for MHC class 1 suppression is MHC class 1 suppression in virus-infected cells or tumor cells. Is this relevant in HPV infections? What is your strategy for those cases? So I think you same I answered those questions.
Yeah, so I think I already answered that. So for HCC, we see a good HLA-A response by staining tissue sections. So in HCC, not so much a problem. However, for example, for cervical cancer, this has been published already that this is a problem, and we have strategies to encounter this, but I don't want to share them today.
And the last question is, had the patients been vaccinated, full HPV vaccine? So I don't know whether this refers to the inclusion criteria for a study or the design of your overall principle of your treatment. So
yeah, so actually we don't even ask for it, because I assume a person that is vaccinated will not get infected and will not develop a tumor. So I'm pretty sure they are not vaccinated. What we have discussed now, but are still ongoing in discussions with the clinicians, is whether we should vaccinate them after the cell therapy, because now they have these very low HBs antigen levels, and maybe by vaccination we can help to induce also an antibody response and at least help them to cure the infection.
Can I ask one last question before we let you go? Basically, for me as a pathologist, it's always the question of fibrosis. Is fibrosis or cirrhosis an exclusion criteria for a study?
We accept until, I don't know if it was Barcelona criteria or Child-Pugh BC.
Okay.
So we look for this, and there needs to be a certain level of liver function that is still maintained, because otherwise we would not want to risk targeting also infected hepatocytes and risking decompensation and liver failure.
Well, thank you very much for your presentation and for answering all those questions. This is very much appreciated, and we have a substantial number of people in the audience. So I think this topic is of high relevance to the community. So once again, thank you very much.
Thank you.
And we are now moving to Dr. Takis Athanasopoulos. I think I get better with your name. So welcome to the lunch talk. And he will introduce Vigeneron. And Dr. Athanasopoulos has recently been appointed as VP of Gene Therapy Research and Vector Technology. He brings over 25 years of experience in cell and gene therapeutic approaches. He has previously held various leadership positions at GSK and has developed extensive experience on various vector technologies. And Dr. Athanasopoulos holds a PhD in biomedical science from the University of London. So welcome, Dr. Athanasopoulos, and we're happy to see your presentations, which I don't see at the moment, but I think you will start them.
Yeah, thank you, Professor Haas. Thank you for the introduction. I'll just start sharing my screen, and hopefully, can you see the presentation now?
Yeah. Yes.
So thank you very much for the fantastic introduction and bringing me to the BioM series lunch. I just want to present our company, Vigeneron, a next-generation gene therapy company. And Vigeneron is a company with 3 novel platforms and lead programs to address high unmet needs. Originally is based in Munich, in Germany, and is spin out of Ludwig Maximilian Universität in Munich, with a team having a strong track record in ophthalmic indications and retinal gene therapy. They are 3 interesting platforms that we manipulated. First is a next generation of novel adeno-associated viral vector capsids. And adeno-associated virus, for the audience that may not be familiar, is a small part of virus, is around 20 nanometers, is extensively used for direct in vivo gene therapy applications. The second technology is called Revert, and this aims to reconstitute large gene fragments more than 5 kb by collating, merging together two AAV vectors via mRNA transplicing. And the third platform technology that we bring forward is proprietary AAV transactivation. That is a mutation-independent technology suitable for mono and oligogenetic diseases. In the pipeline, we have late ocular programs. One is the VG901 that targets retinitis pigmentosa and is currently under CTA and IND filing. And the second one is the VG801 that targets Stargardt disease and is currently at IND enabling studies. There are other also programs for wet AMD. From an investor's perspective, we are backed up by investors like WuXi and Sequoia Capital, and also there are other partnerships that we have established strategic collaborations like with Regeneron. There are 4 co-founders of the company. Our CEO, Dr. Caroline Zhu, with 10 years experience at Novartis. She has led global and regional cross-functional teams in various functional areas. She acted also as a senior management consultant at McKinsey and Company and has more than 5 years of scientific drug research development experience at Columbia University and Technical University in Munich. Our professor, and perhaps a lot of you know Professor Martin Biel, is our scientific co-founder and scientific advisory board chairman, is a full professor and chair of pharmacology, more than 20 years of experience in disease mechanism. He has discovered the CNGA3, and he brought successfully to the clinical stage with more than 200 original publications. And of course, he's a member of the German National Academy of Sciences. And coming out of Professor Martin Biel's laboratory is Professor Stelios Michalakis, a fellow Greek and a professor for ocular gene therapy in the Department of Ophthalmology with a strong track record in retinal gene therapies, and a recipient of DFG, NIH grants, and an author of more than 100 original publications. And Professor Elvir Besirovic, that is now a full professor of experimental and translational ophthalmology in the University of Zurich. And his expertise is within the disease mechanism. Actually, he was the pioneer of the discovering some of these platform technologies that we will analyze further down our talk. Our team has a strong track record, capabilities, and technology in various aspects of AAV gene therapy vectorology, in the vector design, optimization, evaluation, and selection of promoters, in vitro potency assays, efficacy studies, GMP and GLP toxicology, agency interactions with AMA, FDA, and of course towards phase 1 and 2 that these programs are aiming to. So it's a well-versed team. And a few more words about the technologies. So there are 3, as we mentioned, technology platforms. The first one addresses the limitation of limited transduction efficacy using conventional adeno-associated viral vectors. So we have engineered, and I will introduce to the team the way that we have engineered a particular serotypic capsids that are characterized now with higher diffusion properties, transduction efficacy, and excellent lateral spreading, and also reduced immune response. The second technology, the Revert, addresses the limiting packaging capacity of AAVs, that is less than 5 kb. So for genes like ABCA4, that is the gene that we target for Stargardt, is around 6.8 kb in molecular size. I think is this useful technology. And of course, the third technology, AAV transactivation, is through regulation of CRISPR-Cas9 mediated AAV gene therapies, and has the potential to co-regulate multiple genes and knock down or activate simultaneously. So a few more slides on the respective technology platforms. The first one, the VGAAV platform, and how we engineered novel AAV vectors with high transduction efficacy. The rationale was that when you target different ocular diseases, you have different routes of administration. So you can have, let's say, intravitreal injection that is less invasive, but in a sense, and it's not so efficient as a way because you diffuse the vector. You have subretinal that you administer at the back of the eye, subconjunctival, supraarachnoidal. So you have different routes of administration, but in order to pursue the intravitreal route of administration, you need certain AAV capsids with high diffusion properties. So we had to engineer and maintain novel AAV capsids with support efficient transduction via this route. And the way that we engineered this constructs that we published this study in Nature Communications and EMBO journals. In a sense, we created the library of constructs. Variants were displayed through a peptide display library. So in silico screening of a library, we created a 7-mer insertion. For the people that are familiar with the topology of AAV, it has 3 regions. It has a VP1, VP2, VP3 region. So we put an insertion of a 7-mer in the VP1 region. And then we selected very rapidly, within 24 hours of selection, and through different rounds, the most promising capsids. And we identified 2 novel AAVs. We call them AAVGL and AAVNN, with superior transduction properties, in particular in retinal cells. But also we have identified a range of other AAV pseudotypes that are applicable to other tissues. For example, if you have applications targeting the heart, skeletal muscle, pancreas, and this is work in progress. For our main ophthalmic indications, novel AAV vectors show superior intravitreal transduction efficacy. And we have tested this originally in the mouse model. So we put a dosage of 2 × 10⁹ vector genomes in 1 microliter, intravitreal injections in adult mice. Here you can see an example of a cassette that expresses the EGFP under the transcriptional control of the CMV promoter. And you can see that through time, so this 3 weeks post-injection, you can see that all the tissue is expressing GFP, and also the layers of the ocular that we are more targeting. So let's say the photoreceptors, the nuclear layer is targeted, but also other layers like the inner nuclear layer or the ganglion is targeted. But you can see here also, and through antibodies, that the technology is very efficient, and the GL and NN capsids efficiently target, let's say, photoreceptors, in contrast to the wild-type AAV2 vector. So that was promising in a small animal model of disease. And then we went to higher models like the dog model. Again, single intravitreal injection. We increased the dosage to 2 × 10⁻11, again CMV. And similar data have been obtained in the dog model, targeting the photoreceptors. And in the non-human primate, here you can see examples of cynomolgus monkeys that they received 10⁻12 and the same constructs. And again, let's say the same pattern of expression, but also closer to the human clinical stage. We tried into human retinal explant and organoids, and we had the same pattern of expression out of this highly versatile and diffused vectors. And you can see some images here of epifluorescence or co-localization confocal images that they demonstrate the efficacy of the vectors. So as a small summary of this first part of novel generous engineered AAV capsids, we have generated some proprietary AAV vectors that efficiently can cross biological barriers or other tissues. There is excellent lateral spreading that may result in superior visual outcomes. They have high level of gene expression. They can be targeted through intravitreal administration that is less invasive. They work well across multiple species and are capable of transducing both human rod and cone photoreceptors. We can produce this at a high yield, so from a production and processing, these vectors are really good, and also partially evade neutralization antibody responses because we diversify perhaps the interaction with the original heparan sulfate proteoglycan receptor. So this all very interesting. In the second part of the talk, I want to refer this technology platform is called the Revert, where we combine two AAV vectors in order to increase the cargo and to deliver large genes via mRNA transplicing mechanism. And the principle is very simple. Here, you can see, for example, in this organogram, the molecular construct of AAV vectors, and they are flanked by inverted terminal repeats. And after you have the promoter, the first part of the gene of interest, and then you have splicing donor and binding domain, followed by a second vector that has a similar structure. Again, the promoter, the binding domain, splicing acceptor, but also now you have, let's say, the poly of origin. And if you combine the two vectors, you end up with an mRNA that combines the two lateral parts. So the gene of interest is split into two vectors, depending on the size and on the cargo, but afterwards it's unified together, and the protein of interest is assembled. So it's in principle, a very interesting concept. And we have tested this one through different reporter genes. And you can see here luciferase, where the luciferase was split into two, and afterwards was administered into different mice models. And here you can see three. Of interest, actually, the heart seemed that after intraperitoneal injection into the wild-type mice seemed to accumulate the majority of luciferase across species. But also, this one was tested in human retinal organoid transductions, and you can see how it compares against a plasmid that carries the full length sequence, and how the revert sequence of two vectors, let's say, is bringing at very good percentages of efficacy that two spliced vectors together. So we tested this one, this principle in vitro. And also offers multiple advantages. Now we have validated the technology through different reporter genes. We used cerulean, GFP, luciferase, but also different target constructs, MIO7. And now we are, of course, focusing on the ABCA4. Is applicable to various tissues. Is validated through, is independent of serotype, because AAV has different serotypes. So we have tested this one with AAV2, AAV8, AAV9, different routes of administration. And the advantages is that it's an efficient technology. It's very flexible in the splitting side. So you may have heard of split inteins. There the design is more laborious, but also you have like a bacterial protein remnant. So this is advantages of our technology, because you have no artificial remnants. So it's relatively safe, doesn't generate potential immunogenic responses. And the third part is AAV transactivation, where you can regulate one or multiple genes simultaneously. It is based on the previous concept. So you can have the novel AAVs that are combined with a dual system, and you can have, let's say, a DEDCAS9 SP protein that can activate homologous genes. So if you have, for example, as an example, you can have the Duchenne muscular dystrophy patients that are dystrophin deficient, and you can activate the eutrophin homolog or paralog of a protein, but also in other genes, multiple gene activation. And the idea is similar, but you split the dCas9 into different genes, but also you can use different and multiple sgRNAs. So the advantage of this technology is that you can regulate simultaneously either one or a number of endogenous genes. It can be applicable to multiple hereditary disorders, and transactivation can potentially be used for the mutation of one or coactivation of different neuroprotective factors, but also can be applicable to a range of tissues, such as retina, brain, heart, and skeletal muscle. So just to summarize, now we have an in-house product pipeline. We have 3 different platform technologies, but also from an asset, we are targeting more as Regeneron the inherited retinal disease space, and we have 2 products. The VG901 that targets retinitis pigmentosa, and the population is around 8 to 10,000 US EU, and the VG801 that targets the more common Stargardt disease, but has this large gene, the ABCA4, that's 6.8 kilobases, and is around 150,000 in US and EU. The first product is under IND enabling studies, and we have submitted protocols into Paul Ehrlich and I think to EMA, and the next stage is FDA submission. And the second one is just a pre-IND enabling studies. Just to mention a little bit more about the 2 diseases, the CMG1 is a gene therapy for autosomal recessive retinitis pigmentosa. That's a group of inherited retinal disorders. It's an unmet medical need. No therapies are yet available, and the disease progression results in loss of night vision, progression tunnel vision, and lifelong visual functional decline. And these are some of the attributes of our product, with our approach being superior, with a novel AAV vectors can submit a broader transduction of photoreceptors through an intravitreal injection. That will be a first in class, and we are preparing towards the clinical stage. The second program is the VG801 for Stargardt disease. Again, Stargardt is a recessive inherited eye disease due to mutations to the ABCA4 gene that accounts for almost 95% of Stargardt disease progression. There are no approved treatments, and again, results in vision deterioration, bilateral central visual loss, and combines the 2 attributes and the novel capsids that we have engineered, but also the revert technology that you can put through mRNA transplicing the 2 parts of the gene and bring them together safely into the patient population. So that's it for us, Regeneron, next generation gene therapy company. Of course, if you have any questions, and I just want to conclude with a final slide of the team. These are the people that they do the work, and our Regeneron scientific team at Planegg site. And you can see here the scientists, Vicky, Verena, Michi, Patricia, but also all the research associates, postdocs, directors, lab technicians. Some of those because it was a photo taken yesterday in Planegg. So I would like to thank you all for your attention, and I invite the audience for questions. Thank you.
Thank you very much, Dr. Athanasopoulos, for this very interesting presentation. I mean, I've been in gene therapy many, many years back, so I'm happy to see more and more progress in this field. And there are actually 2 questions already from the audience. The first one is, did you produce the virus or which yourself, or which CDMO did you use to engineer the capsids?
A very good question. We internally produce the virus in small amounts, so we have viral vector production capacity and under SOPs. But for large scale production and for the clinical stage, we will be using most likely WuXi as a CDMO. So for small scale internally, for large scale, we may outsource to a CDMO.
Thank you. And the next one is, what are the therapeutic applications for your revert technology? I think you mentioned this, but maybe you can comment on this again.
Of course. So a very interesting question. The applications are limitless. They target mostly large genes. So genes that are not into the cargo capacity of a single adeno-associated virus as a single administration direct injection. So it can be genes of large capacity. You can bring 2 or more vectors together through mRNA transplicing. So any disease that has functional genes that they have to be substituted, or through CRISPR-Cas9 activation through multiplex gRNAs are applicable to this technology. AAVs and the novel AAVs can target differentially and specifically lots of tissues. So you can have, let's say, an AAV9 that can target the musculature and also the heart very efficiently. You can target liver, and you can check, let's say, the different transaminases. So in a sense, you have to think of what is the gene that you want to replace. If it is of a certain cargo that exceeds the normal cargo capacity of normal vectors, then this is a technology for you, and we would be delighted to collaborate.
Thank you very much. And I have one or 2 questions. So why did you choose the eye? Because you think it's a great target, because it's a great target population, or are there actually safety reasons why you would like to, let's say, a very limited organ space?
Very good question. I guess it's a combination of factors. One is the expertise of the team, the existing expertise of the co-founders. They are pioneers in the ophthalmic disorders, and of course, they have collaboration with expert clinical team. So the setup of the team is one factor. From a biology perspective, the eye is a relatively immune privileged tissue. Is concise. You have to think that for organs that they may need access through systemic administration, you may cause unwanted immune responses. That, of course, they are manageable. There are different ways through steroid or different parts of TLR9 pathway inhibition that you can ameliorate. But I think that the eye offers a space that you can apply this technology in a far safer way for this indication. But also the other extra advantage is that you don't need, from a process development perspective, so much vector. So if you go systemically, and especially for Duchenne muscular dystrophy patients, for example, that they have a muscular disorder and you need to target 40-50% of your body mass, then you may need to exceed 10 to the 15, 10 to the 16 vector genomes in order to go systemically. For the eye, you need a relatively low dosage. That means that it's more immune beneficial in a way, but also from a process development perspective is of low cost. So these are some of the reasons that we target the eye.
Thank you very much. And there's another question from the chat. Are your VG-AAV capsids patented?
Yes. So there are 3 patents that they cover the 3 platform technologies. So the GL and N capsids are patented, and there are a family of patents also that they cover other derivatives or other variants of these serotypes. They have submitted applications, I think, both locally and at the European level. Yes.
Thank you. And let me allow a personal last question. Are you planning to do your clinical trials in Germany or even better in Bavaria?
Yes. So as far as I know, the targeted population of the CNGA1 asset with the advice of from Paul Ehrlich Institute will be will be in Germany. So I think Germany and Europe will be the first patients that we will be accepting.
Well, I'm very happy to hear this compared to unfortunately, other clinical trials that moved to outside of Germany. So thank you very much. I think there are no more open questions to both presenters, Dr. Viskirchen and Dr. Athanasopoulos, for those great presentations, for the audience to attend this lunch talk series and for asking those questions. And I'm very happy to see you or hear you during one of the next lunch talks. And let me repeat what I said 12 years ago. Good luck to everybody to move forward. So thank you very much and have a great afternoon.
Thank you, Professor Haas. Thank you.